हम्म? क्या? हाँ, प्लांट ब्रीडिंग का डाल।
ते मध्ये ते छोटं लागतं ना, मी बघतो, ट्रॅक करतो तुमचं लोकेशन. सावकाश लोकेशन तुम्हाला दिलंय ना, लोकेशन लावा. आ गए. व्हाट्सएप मी माझ्या मी सांगतो काय होतं. किलोमीटर आपल्याला तुमचं पुढं आहे ना, आता कुठे हा बायपास आहे.
Good morning. Good morning, Dr. Apurva.
Good morning.
Good morning, Dr. Tikoo, sir.
Good morning. Good morning. Not seeing you yet, but good.
Yeah.
वीडियो ऑन करिए. काफी दिनों बाद मिल रहे हैं आपसे. How are you?
Fine, sir. Fine.
Good, good.
Seeing after a long time, sir.
Next time I'm in Banaras, we must meet.
Good. Yes, sir. Sure, sir.
Yeah, I was there in October last.
A program over here.
बनाएंगे program.
नहीं, मैं तो रहता बाहर में हूँ, लेकिन BHU से एकदम सटे ही है.
अच्छा.
ये करौंदी. जी.
Yeah.
ऑफिस में है.
But many of them probably join on Facebook or something. Or what? अभी 11 हुआ नहीं. हाँ हो गया. Maybe couple of minutes more. अभी suddenly ऐसा और join. Some people are joining. I see. We have to find वो Facebook, YouTube वगैरह से कौन ज्वाइन करता है. क्या हो गया? I hope you are circulated among the university people. That's all. Did you circulate it within the university?
Yes, sir. I have circulated it.
Total कितना था registration राहुल? 400?
64.
Okay. Yeah, we have touching 500 registration.
Yeah.
But here it is not really good to go. So, well, anyway, shall we start or shall we wait for?
I think we should start.
Okay.
There are 21. We should not.
Yeah,
wait.
people will continue joining the session. So let's get started. Very good morning, everybody, and welcome you all to this 41st webinar of Plant Breeding webinar series. These webinars are being organized by the Foundation for Advanced Training in Plant Breeding on a monthly basis. And today we have Dr. Kartikeya Srivastava, who will talk about strategies for breeding heat stress in crop, taking the case studies of tomato, Rapeseed-Mustard. The purpose of these webinars are to sensitize people to know and value the importance of plant breeding, and also to create an awareness on various aspects like technology integration, policy support, regulatory overview, farmers' comprehensions, and various other aspects required for inclusive development of crop improvement. Foundation for Advanced Training in Plant Breeding, abbreviated as ATPBR, is a non-profit organization dedicated for capacity building in agriculture, particularly in the field of plant breeding. ATPBR, with its global food reach and collaborative platform, foster the personal and institutional capabilities in alignment with the current and future agriculture needs. The objectives are to develop sustainable agriculture R&D system that serve farmer and support emerging entrepreneurs through advanced research and training, and to assist the seed industry in building market-driven competencies in human resource, making crop improvement an attractive and profitable business. ATPBR conduct professional training, monthly webinar, national and global conferences, and support publication. To date, we have organized 8 conferences, 34 professional trainings, 40 webinars, and reached more than 20,000 students and professionals across the Ghanaian community through our activities. We have published more than 10 books, and our flag publication is Market-Driven Plant Breeding for Practicing Breeder, published with Springer. We have trained professionals not only from India, but across the world, including Italy, Spain, Brazil, New Zealand, Sri Lanka, Bangladesh, Nepal, Afghanistan, Nigeria, Kenya, Ghana, and many more. Additionally, we have 12A ATG CSR certification, making us eligible to execute CSR project for the industry. Now, I would like to take a privilege to introduce our moderator, Dr. Surinder, an independent director at ATPBR. He has more than 55 years of experience as a breeder, teacher, researcher, and R&D leader with extensive experience both from public and private sector. He held positions of Global Vegetable Research Lead and member of Global Management Committee at Adventa, Global Vegetable Development Leadership Team member and Effect Product Development Lead at Syngenta, Global Bush Tomato Lead at Pioneer. He has been FAO consultant on vegetable breeding and production in Myanmar. And as a breeder, he had bred market-leading hybrid of tomatoes and okra for India. Served on various advisory committee of government and non-government bodies, and published more than 50 research paper and review. Now I request Tikoo sir to please take the floor.
Thank you, Aparna. And I welcome Dr. Kartikeya Srivastava, who is a professor in Department of Genetics and Plant Breeding, Institute of Agricultural Sciences, Banaras Hindu University, Varanasi. He has worked in various capacities in the corporate sector that included Tata Energy Research Institute, Indo-American Hybrid Seeds in Bangalore, and Namdari Seeds, Bangalore. His work on vegetable breeding, seed production, and export involved breeding, strategic research, planning, and management. He worked on the development of cauliflower and cabbage hybrids for India and overseas. Developing synthetic variety of Brassica napus, he developed it as Indam Canola 1. He developed cauliflower hybrid NS120 and one thermotolerant Indian mustard variety HUJM 10-6, which is named Malviya Nidhi and released by UP State. His area of interest: breeding for abiotic stress tolerance in vegetables and oilseeds, developing molecular marker systems and mapping populations for breeding stress tolerance in vegetables. He had a major research project under the University Grants Commission named Genetic Improvement for High Temperature Stress Tolerance in Tomato. He has standardized the methodology of screening genotypes for heat stress in vegetables and identified tomato genotypes and hybrids tolerant to high temperature. UP Council of Agriculture Research has funded project for developing climate resilient Indian mustard genotypes. Dr. Srivastava has published more than 90 national and international papers, review articles, popular articles, book chapters, and manuals in research area. He has produced 18 PhDs, 45 post-graduation dissertations, 1 postdoc, and honored by various societies and national bodies. That's a great CV to have, Dr. Srivastava. So I give the floor to you. Thank you.
Thank you, sir. It is great a privilege for me to share the screen with you and Dr. Aparna. ATPBR, I thank to Dr. Aparna from ATPBR. She has given opportunity to talk on strategies for breeding heat stress in crops. Here I'm taking the case studies of tomato and rapeseed. This is my privilege, and I thank
Welcome.
you all. 1 minute.
You can make the full screen.
Full screen. I'm putting it in on full screen. So,
No, it's not happening. I don't know why.
is it?
Is it a PDF?
No, it is PPT.
Then you should get the full screen on it somehow.
Okay. 1 minute.
Maybe you have to exit and do it again. Or
1 minute. Sir. I'm making it full screen now.
नहीं वो नीचे है ना icon. TV icon at the bottom right. Oh, you exited? फिर से share करिए.
In the meanwhile, good morning, Tikoo sahab.
Morning. Good to see you after a long time.
Yes, I am seeing you. And it's a privilege to see Kartikeya today after long.
Yes, sir.
BHU जाके तो मिलना नहीं हुआ, लेकिन यहाँ मिलना हो रहा है.
ऑनलाइन पे मिलना हो जाता है सर.
चलिए.
सर.
क्यों नहीं हो रहा है ये?
Now, is it visible in full screen?
No.
Yeah, it is actually, it is seen, but on the side, it is not actually the full screen.
It's not full screen. आप जरा ऊपर जाके वो स्लाइड में स्लाइड शो करिए.
टॉप में जो है उसमें फुल स्क्रीन वाला करना पड़ेगा. टॉप में
टॉप में ट्राई करिए.
टॉप राइट में
Okay, sir.
फुल स्क्रीन.
नहीं आपके ना होगा वो है ना क्या कहते हैं स्लाइड शो. गो टू स्लाइड शो ऑन टॉप.
हाँ,
ऑन द रेड बार स्लाइड शो है ना.
हाँ.
यहाँ उस स्लाइड शो को क्लिक करिए. कैन यू क्लिक ऑन द स्लाइड शो ऑन टॉप ऑफ योर स्लाइड. रेड बार है ना ऊपर.
रेड बार है.
या
Yes.
ऊपर स्लाइड शो लिखा है ना. ऊपर रेड बार है. ऊपर रेड कलर्ड बार ऑन टॉप ऑफ योर above the font and वो है क्या paragraph or something. There is a slide show there, no?
Yeah, slide show is there.
हाँ उसको क्लिक. अब full screen. तो full screen नहीं आ रहा. Start slide show.
Yeah.
From beginning, sir. From beginning.
Press on from beginning.
Okay.
वो लेफ्ट में है आपके.
Slide show
Yes.
from beginning. Yes.
उसको क्लिक करिए.
Click here. Is it visible now?
नहीं नीचे स्लाइड शो भी है ना. उसको क्लिक करिए. ऊपर है ना. फ्रॉम बिगिनिंग के बगल में नीचे स्लाइड शो लिखा है.
From current slide भी या तो भी करें.
करंट स्लाइड के नीचे
नहीं हो रहा है सर. आई थिंक कुछ तो इशू है.
आपके पास है क्या वो slide?
नहीं सर, वी डोंट हैव इट.
Can you go to the next slide?
Yeah, it is on next slide, sir.
No, but it's not moving. That's why. आप exit
Yes.
करके फिर से करेंगे, please.
ठीक.
now we can see the next one.
Yes. Now we
Yeah,
can
now we can see the.
third
But
one.
we didn't see. This one we can see. Yes.
Yeah.
Okay. Let us go ahead. Please go ahead. Sorry, these technical glitches.
So, thank you all
Hmm.
for being here. And now, as you know, that tomato is most popular and widely grown vegetable in world. And owing to its fast and wide climatic adaptation, it is universally treated as a protective food and cultivated throughout the India during Rabi season, and also in some pockets where we are having the virus tolerance as well as heat tolerance. It is also cultivated in Kharif, and especially in hills, it is cultivated in Kharif. There is a growing demand for tomato-based product driven by health conscious and convenience. Top 50 tomato processing companies processed approximately 30 million metric tons in 2024, accounting for about 2/3 of the global volume. and optimum temperature required for its cultivation is 20 degrees centigrade to 24 degrees centigrade. And for fruit setting, it is 15 to 20 degrees centigrade. And ideal temperature for color development in the tomato is 18 to 25 degrees centigrade. And for lycopene production, it generally drops down in above if temperature reaches about 27-28 degrees centigrade.
Yes.
If we see the data, the China is the first, followed by India and USA, Turkey in the production, global production, and
Can
India,
you-
the Madhya Pradesh, Andhra Pradesh, and Karnataka, are, Maharashtra are the.
Next slide.
This is the fourth slide.
No, actually, that's why the problem is we are still seeing the second slide, global
Yes.
perspective.
Okay.
Yeah.
I think
Yeah.
what we do is you click on the sidebar on the slide you want to show. Click there, then it will show.
Okay.
आपका left में बार है, जहाँ
Yes.
slides है. Can you click on the slide you
Yes,
want to
yes.
show?
Now we can see, sir.
Yeah. So click there. By arrow, it is not moving. You click on the slide.
Yeah.
Thank you. Yeah, please go ahead.
Okay. So, what is the effect of heat stress on tomato and how the plant responses to the tomato to the heat stress? So, primarily in it's a winter crop, so vegetative, reproductive, and all phenophases of the tomato is adversely affected by increase in temperature. If the temperature goes beyond 30 degrees centigrade, the flower setting and flower drops and all takes place, and affect the high temperature affect adversely both vegetative and reproductive phase, and majorly the physiological processes like your photosynthesis, photosynthetic translocation, cellular membrane organization is affected, pollen viability and pollen germination is affected, stigma exertion in high temperature you find exerted stigmas, and corolla tip burning and fruit set is poor. Corolla tip burning is there and fruit set is poor. So it results in inferior quality because of the lycopene synthesis is affected. So patchy color development occurs and fruit size becomes small, and generally low yield is obtained. And you find in summer the patchy tomato, it is only because of the high temperature and lycopene synthesis is affected. So if we talk about this thing, the effect of heat stress. So pre-anthesis temperature stress is associated with the developmental changes of anther. In anther lobe, there are the epidermis, endothecium, and tapetum. So because of this, the irregularity in epidermis and endothecium is there, and the tapetal layer is also affected because of the high temperature stress, and pollens are these tapetal layer are feeding the pollen, developing pollens. So the pollen sterility is the resultant, and because of this pollen sterility, the fruits are not formed and flowers are dropped. And number two, if the parthenocarpically the fruits are formed, the fruits remain small. So high post-pollination temperature inhibits the fruit set. Fertilization is sensitive to high temperature stress. There is stigmatic surfaces which goes dried, and the pollen germination does not take place, and because of that the fruit set is affected. And there is significant inhibition of photosynthesis occur at a high temperature above optimum, and resulting in considerable loss in potential productivity, And you know, the photosystem second is highly sensitive, and because of that, the photosynthesis is affected. The carbon fixation is affected, and apart from this, there is the membrane sensitivity is there. Membrane thermostability is affected. So it is there. Then progress in developing heat tolerant cultivar has been hindered by complexity of the trait and low heritability values. So here you can observe the heat tolerant lines and heat susceptible lines. Heat susceptible lines, the fruits are not turning yellow, not turning red because of the lycopene synthesis is affected, and fruit set is poor. Whereas heat tolerant under high temperature stress condition, they are setting the fruits. So we conducted an experiment during 2010 and 2009 onward, and we had the temperature regime right from increase right from March onward, and it is going up to 40 degrees centigrade, and the diurnal temperature and night temperature is very crucial. Here, the night temperature, if it is above your 32 to 30 degrees centigrade, the pollen viability is affected. In second year also, we had the similar temperature regime. After March, 30 degree onward, and it reaches in April, that is 38 to 40 degrees centigrade. And we carried out a screening in 2009, around 72 tomato genotypes, and out of which we have taken out 34 for further screening. And it is mentioned over here, that is Pusa Sadabahar, TBRT1, H8874-1, Kashi Amrit, Fluoridate, Kashi Sharad, DT2, Pyant T3, H24CO3, Punjab Upma, and likewise 34 genotypes we took in under screen. And we first, since the pollen is affected because anther is exposed outside, the pollen is affected. So we took the pollen germination in vitro condition. We made a pollen germination media following the Bedinger's in vitro liquid tomato pollen germination protocol, and we measured the pollen viability. We took the crushed the anther and took out the pollen and observed this thing. Here you find two varieties which are showing in Floridate and Pusa Sadabahar at a very high temperature, that is around 40 degrees centigrade. We took it in a growth chamber as well as in Floridate and this thing. So it has shown a differential response to the pollen viability in heat stress condition. Here you see FLA 7171, which was showing a good pollen germination. Here the pollen viability is also very good, and same way the BT 120, it is showing less viable pollen because acetocarmine, it is treated with acetocarmine, and it is getting less color. The pollen is taking up less color, so they are more non-viable pollen. So pollen viability response we took in crosses also with the selected parental line. So Pusa Sada Bahar cross class 7171. Both are the heat tolerant, and you observe a good pollen viability in this hybrid, whereas selection 7 cross flower 7171, it is showing a moderate pollen viability, and Kasi Amrit cross superbag is showing a lesser pollen viability. So this is only because a differential pollen viability responses. Now, this is has been observed in the fruit set in tomato hybrids. The Pusa Sadabahar H8874-1. This has given a good size tomato under heat stress condition, whereas sun cherry, by virtue, because cherry tomato is having proliferous fruit bearing, sun cherry cross superbug. Superbug is both are the heat tolerant lines, and their cross has given a good fruit set under high temperature stress condition. And similarly, we take the stigma exertion as a selection criteria for heat stress tolerance. So one way it is a heat tolerant, and other side the heat susceptible, where the stigma is protruding outside too much, and because of this the receptivity of stigma is lost, and the flower drop occurs. So if it is not receiving pollen and fertilization is not taking place, so here you see the stigma exertion, and same way the flower drop in the next slide. Number of flower dropped upon the total number of floral bud is taken into consideration for calculating the percentage. So flowers are dropped at in several cases at a high temperature. There is one selection criteria which is very much taken up for estimating most of the crops, especially in tomato as well as in rapeseed-mustard also. That is membrane thermostability. We take the this leaf disc from the tomatoes under normal condition and heat stressed condition, and take out the electrolyte leakage during the high temperature, and we measure it this the more electrolyte leakage in the outside the cell, and those which are showing more stability in the membrane, and there is less fluidity, and they are more stable in nature. And this denaturation also occurs at the protein which are embedded in the membrane, and these, if they get denatured, the protein function, they are the functional protein and responsible for signal transduction and ion exchange, selectivity of the ions across the membrane. So if these membrane bound proteins are denatured, their functionality is also disrupted. So, therefore, the membrane integrity is a key parameter, and one must look into this. And other say heat sensitivity characters for screening tomato is corolla tip burning, percent fruit set, number of seeds per fruit, percent seed germination, and quality traits like your lycopene content, total soluble solid, and titratable acidity is also need to be measured, because the quality of the tomato is because of these. And we found that Pusa Sadabahar, one which was released from IARI for cold set, and it is also performing good for hot set condition. Same way, FLA 7171 here and NDTVR 60 selection 7, they have shown considerable pollen viability, pollen germination, and relative cell injury under these conditions. So they have been considered as a tolerant genotype. So here, if you see across the trait, flower drop percentage, pollen viability, pollen germination, cell membrane injury, and fruit set and average fruit weight, you find that Pusa Sadabahar, FLA 7171 are showing for most of the trait, followed by your NDTVR 60 and selection 7. So these can be taken as selection criteria and for the heat tolerant tomato. And we made the cross for genetical studies in line tester analysis, as well as we have also gone for the generation mean analysis. And these were the parent. Some were the lines, like your fluoridates, NDTVR 60 PS1, Pusa Sadabahar, and these were taken as the line, and the tester were FLA 7171, H88-74-1, and Superbug. So here you find the Pusa Sadabahar, which is forming the fruit even at 42 degrees centigrade in Varanasi condition, whereas at the same place, the fluoridate is showing you the flower drop and no fruit set at all at a higher temperature. So for the genetics of the heat tolerance, we have taken these 6 traits for study, and various procedure like your TTC staining for pollen viability, pollen germination, that under pollen germination medium, and corolla tip burning is taken as percentage, flower stigma exertion is taken as percentage, and membrane stability as mentioned by electrical conductivity method, and fruit set were taken. And the genetics has revealed that most of the characters are governed by the quantitative character, having epistatic character, or dominance component is there. And some of the characters like corolla tip burning is a complementary gene action, and duplicate gene action for stigma exertion, and also for the pollen viability, the pollen germination, membrane thermostability, and fruit set, the duplicate gene action have been studied. And it is suggested that if we go for hybrid breeding, we can exploit these gene actions, and we can have a good hybrid. And we also calculated HSI, that is heat susceptibility index, on the yield parameter, And the role of pollen starch has also played an important role, and a soluble sugar content in tomato. So our first physiologist, Professor Praveen Prakash, and his student and myself, they have conducted this test, and we took the four varieties like Pusa Sadabahar, NDTVR 60, and 86 and fluoridate for relative tolerant and heat susceptible varieties, and anthers were collected at 10 flowers, were dissected according to the Auli et al., and pollen grains were obtained by slicing anther and transversely and vertexing them in cold sucrose-free germination solution. And it was observed that carbohydrate concentration on the pollen sample were analyzed, and soluble sugar were determined by HPLC, and fast carbohydrate column of BioRed has been taken, and detection by refractometer was also done. Findings show that the high temperature is strongly disturb the carbohydrate metabolism in pollen grain in tomato, and optimal temperature condition is 28 degrees centigrade, 21 degrees centigrade, that is day and night, and resulted in starch accumulation in the pollen grain, and where it reaches maximum value before anthesis and diminishes towards anthesis. So, if the starch accumulation is more before anthesis and soluble sugar at anthesis, the pollen viability and the pollen germination is more, and this provides the pollen grain with an instant energy source for their germination. The starch and soluble sugar content in the pollen grain is most important biochemical parameter for screening tomato genotype for improved fruit set under heat stress condition, and Pusa Sadabahar and NDTVR 60 has showed the can be used as a source for heat tolerance for further study program. Same way, we conducted the experimentation on rapeseed-mustard, and the rapeseed is an important oil seed crop grown in northern and NEPZ, northeastern plain zone, and here we are having the four indigenous species, that is Thoria, that is Brassica campestris or Brassica rapatoria, Brassica, there's brown sarso, yellow sarso, and Indian mustard. Apart from this, we are having Brassica napus with us, which is being cultivated in Haryana, Punjab, and Himachal area very much. And the optimum temperature for this is 6 degrees centigrade to 27 degrees centigrade, and good in annual precipitation of 50 to 420 centimeters of annual rain precipitation, and production is majorly constrained by high temperature and erratic rainfall. Here in Uttar Pradesh and Bihar, which is a majorly paddy growing area, here the paddy is sown late, so transplanted late, and that's why the late harvest is there, and the land is free by end of October or mid of November. And if you sow the rapeseed and mustard under late sown condition, it is forced to fall under terminal heat stress. And also those farmers which are sowing early, they are also subjected sometimes to early heat stress under field condition. So this terminal heat stress under Rabi season crop, it is a major constraint for heat stress. And if you see the productivity, there is a rising trend in the productivity of rapeseed and mustard, and we have surpassed this year the soybean and become the first in the production and productivity in whole of the oil seed, and this is only by virtue of newer variety development. Those are having abiotic stress tolerance and biotic stress tolerance. So if you see the impact of the heat stress on the plant growth, it is at various level, that is physiological, biochemical, and yield level, productivity level. It is because of if you see the physiological level, the reduced leaf water status at a high temperature, dehydration, increase in transpiration, and impaired photosynthesis and chlorophyll degradation is there. Reactive oxygen species are produced, and accumulation of stress metabolites are there, and there is limited phytochemical efficiency. There is degradation in chlorophyll, as well as the photosystem second is being affected, and most of the reactive oxygen species like your singlet oxygen, your superoxide development, and this is H2O2 development. So these are responsible for reactive oxygen species production, and because of that the cellular damage is there, and this cellular damage limits the various photochemical efficiencies, as well as membrane integrity is damaged. So reduction in leaf development is there, reduced plant growth duration, increase in leaf senescence is there. If there is increase in temperature at terminal stage, the plant is subjected to mature early, and because of that the yield is affected. So if the temperature is early temperature is there, the reduced seed germination and seedling vigor is reduced. Poor stand establishment is there, and less number of siliqua per plant. And not only siliqua number is reduced, seeds per siliqua is also affected. And this is only because of the pollen viability and aborted flowers are there. The poor grain filling rate and grain quality is affected. So same way as we have done in earlier tomato experiment, we go for the pollen viability test and using acetocarmine method. And here we did the pollen viability. We screened out those genotype which are having a good amount of pollen viability, and that is, as I have mentioned earlier, it was a selection criteria. Here we have added one more trait, that is relative water content under heat stress, because in rapeseed mustard, the heat stress is also having confounding effect with the like drought, and relative water content becomes
It's not possible because we have already taken dimensions to the level number 2.
So
Can I request everybody to mute your system?
Mute your system, please.
okay. So here the method is followed as Barr and Witherley in 1962 has given, and we place the sample for relative water content under this thing for its full turgidity, and we oven dried it, and we measured the dry weight and the fresh weight, and on that basis we calculated this thing. Same way, the membrane thermostability is also a important criteria, and as mentioned by Sriram in 94 and Shanan in 1990, we followed this MSI membrane thermostability index on the basis of the conductivity meter test, that is those which have shown a good electrical conductivity, those are sensitive, and those which are having lesser electrical conductivity, those are the tolerant genotype. So this is the way we have calculated the electrical conductivity, and treatment was given. Then seed germination and seedling vigor. This was done under growth chamber condition, where we have maintained the temperature, the normal at 30 to 32 degrees centigrade, and elevated temperature 42 to 44 degrees centigrade. And we measured the germination in both, and germination percentage was calculated, and vigor index was also calculated on the basis of the standard germination percentage into seedling length. Here we conducted this under growth chamber, and we found there is a variability regarding the seedling vigor under heat stress condition. And same way, seedling biomass from these, it has been calculated, and seedling biomass as a photosynthesis translocation, as well as reserve in the seed, in the sperm, how it is going to give the seedling biomass. This is also being studied. Chlorophyll stability index has also been calculated with the SPAD meter in the field condition, and this has given a good this thing criteria for selection for the terminal heat stress tolerance. So we have taken this, and apart from this, we have also gone for the canopy temperature deficit. That is because those variety which are able to lowering the canopy temperature, they are also considered as a tolerant genotype. But there are many more parameters which need to be included in this. And here, what we screened out, here we have taken around 85 genotypes, and out of which we taken out some of the genotypes which were good heat tolerant, and with the various parameters like your RWC, MSI, and pollen viability test, and chlorophyll stability for various first and second. This has turned out with the some of the genotypes like your Azad, Mehak, Vaibhav, Usatarak, Basanti, and BPR 543-2. This is a variety from, well, not variety, a line from Bharatpur, which has given a good this heat susceptibility indices Vardhan HUGM 10-6. This is HUGM 10-6 is one which has been released as a Malviya Nidhi recently, quite recently, and RLM 13-49 from this Punjab Agriculture University. These are the varieties which have shown a good parameters, physiological parameters, and they were considered as a tolerant genotype. And heat susceptibility indices has also been calculated, and it is with the formula given here. And HSI, which is less than 0.5, were considered as a tolerant, and moderate, which is between HSI 0.5 and less than 1, and more than 1 is considered as susceptible. And same way, heat tolerance efficiency was calculated, and those which are having higher heat tolerance efficiency were considered as a tolerant genotype. So here you see the mustard genotype promising under heat stress condition at Varanasi center. So we find that BPR 543, Vardhan and Basanti are having a good yield. HUGM 10-6 is also having good yield. Vaibhav is having good yield. So they are having potential to produce more under heat stress condition, and around more than 15 quintals per hectare yield is obtained by these genotypes. Same way with the HSI, those which are having lower value, you see that PPR 543-2, Vardhan and Basanti, as well as Ajad Mehab, are having the lowest HSI, means they are having higher heat tolerance. So generation mean analysis has also been conducted by one of my students. It has shown that this yield per plant is showing additive into additive interaction, and complementary gene action for across 14 different traits has been observed. And among high interaction crosses, Vardhan cross PM25 has showed a strong additive into additive interaction for days to flowering, biological yield, and additive into dominance interaction for oil content. And dominance into dominance interaction was found in Kranti cross PM25, base to maturity, primary branches, siliqua length, and biological yield has been found with dominance into dominance interaction. So if you go trait wise, you are having different complementary, duplicate, and epistatic interaction, as well as you find that it is governed by quantitative character, and it is better to have hybrids or biparental mating with followed by recurrent selection, or dilyl selective mating should be done for producing the homozygous line in later condition. So either go with the hybrids or these breeding methods you can have. And selection for heat tolerant line in Indian mustard, screening for terminal heat stress in Indian mustard genotype was initiated in 2005 in AICRPR Rapeseed-Mustard. And in this NRCDR 02 BPR 54 541-4 RGN 152 RGN 145 and BPR 549-9 were identified based on the reduced seed yield loss less than 30% under late sown condition. The varieties like Kanti, Pusagrani, RGN 13, Urvarshi, NRCDR2, Pusa Mustard 25 and Pusa Mustard 27 has demonstrated a good thermotolerance in seedling stage. So we are having the varieties for early heat stress also and late, that is terminal heat stress also. So one should also go for broadening of the genetic base of the rapeseed and mustard by the interspecific, intergeneric crosses, and some of the genera are having the high temperature tolerance, so we can have the crosses with this, some with the Brassica carinata, Brassica tourniforti, and Diplotaxis, and have shown a high degree of high temperature tolerance. And whereas Synaptus alba has shown sensitivity, but in crosses they are contributing to white rust tolerance, and Brassica napus as such is for alternaria tolerance, and Iruca sativa is for drought tolerance. These can be capitalized, but for high temperature stress, Brassica tourniforti, Diplotaxis, and carinata can be utilized. So there is another development of mapping populations. So you can have for QTL mapping, RILs and nested association mapping, magic population and association panels can be utilized for QTL mapping, and there are some of the group are working on this for terminal heat stress tolerance in mustard, and it needs a precision phenotyping. So generally the phytotron or growth chambers for screening the genotypes under high temperature stress condition, under field conditions like your infrared imaging and this thing with the drones can be also applied, and high throughput pollen analyzer. Since the pollen viability is there, important criteria in both the condition, whether it is rapeseed or mustard or tomato. So high throughput pollen analyzers can be used for evaluating the pollen quality by fluorescent diacetate and PI labeling, and genome-wide selection can also be followed for your mapping the QTLs and SNP discovery. So here the genomic selection or genome-wide selection is based on a large number of markers in a population, and linkage disequilibrium with all QTLs with a polygenic trait like heat stress can be mapped and evaluated. And genome-wide selection tends to be most efficient for pyramiding QTL for different traits associated with the heat stress. Here, genomic selection also we can follow. We can give the genetic value on the training population. We can have the genetic value for each of the markers and we do the genotyping and phenotyping on the training population, and we can apply this genetic value on the breeding population, and on the basis of genotyping itself on the breeding population, we with the estimation of the this genetic value, breeding value of the individual lines from the marker data, we can select the individuals. So this is a genomic selection. In this, we can identify the QTLs which are associated with the heat stress. And apart from this, the genotyping by sequencing for thermotolerance in Indian mustard has been conducted by Sandhu et al. In 2019. And here they have the diverse panel of these genotypes, and the GPS activity was done, and around 18,000 SNPs have been identified, and they were put under GWAS analysis, and from this experimentation, 24 QTL for terminal heat stress has been identified in the mustard. And apart from this, this is my end of this slide. I thank to the contributors and the funding agencies, the ICR, IIBR, Rapeseed-Mustard Research, and BHU for providing the facilities, the UGC and UPCAR for financial assistance, Professor Praveen Prakash of Department of Plant Physiology, Dr. Vijay P. from Department of Plant Physiology, and our students, they have different PhD, like Sunil Kumar, Dr. Surinder Kumar, Dr. Aditi Eliza Tirki, Ms. Sivangi, Ms. Rohit Kumar, and Arvind Patel, who has associated with us, and they have involved in generating large data. Thank you all for patience hearing. I think I am in time in completing my.
Perfect.
Yeah.
Absolutely in time. Thank you very much for a very, very exhaustive work on the heat tolerance in both the crops. And thank you also for not only just identifying some heat tolerance, but also getting us through the mechanisms that you have seen that help in getting seed tolerance. Because sometimes we just see the phenotypic result, but we don't get into the mechanisms that deliver these traits. So you have been, I think, in the forefront of doing this work, and also some of the work on the genotype by sequencing, etc. That's, I think, have you, I don't know whether you have yourself done also, but I saw one Sandhu's work. So they have identified some QTLs. So I have certain questions on that, but first, I think we will take the questions from the audience. There were some hands raised before I then will conclude. There's one question saying, what is that? Will the estocarmine test that shows good results, will it also mean good heat set? What is that question?
Sir, acetocarmine is a viable dye, so
yeah, I know, we know. But he says whether this will mean that it will translate into pollen germination also.
cytocarmine, we take it as a viable dye, so it turns the pollen into
As viable.
coloration. Apart
Yeah.
from this, we are also using the iodine solution for staining the starch. And starch is one, the quantitative staining of a starch is also going to tell you the how much this is viable. And apart from this, the modern technique is the fluorescent diacetate is also being used in pollen analyzers. And there are many viable dyes which are being used for estimating the pollen viability.
Yeah, that should answer that question. What is the ideal sowing window for testing heat set tomatoes in Varanasi?
Yeah, actually, sir, if we are sowing it in October, it is a normal sowing condition.
Yeah.
And if we are delaying the sowing by January and transplanting it in February, so by March itself, the vegetative growth is completes vegetative. It enters into the flowering, and March onward the temperature rises over here. And I was quite fortunate both the year. During March, I have crossed the temperature above 38 degrees centigrade. It was reaching to 40 degrees centigrade, and April it was in 42 degrees centigrade in Varanasi. So it was an ideal this thing for doing this.
Yeah, I think that's true. Like in North, in say Gurgaon or Karnal, Ghaziabad, also because of the slightly better heat tolerance in some of the hybrids, they are able to now sow in January, transplant end of February, or even transplant early March, and then you get into that high temperature. And some of those hybrids doing well, they get good price. And I think the mustard also must be the same situation, right?
Same situation is there.
Yeah.
And it is for Uttar Pradesh and Bihar, and those which are paddy growing area. There, our mustard crop is falling, forced to be sown in November, and there is fall in yield because of that.
Yeah. And also, this have you tried the released hybrids that are doing well in that time? It might be a good idea to study.
Sir, actually, we have to give this material to IIBR for this thing. Since we were not a partner in this directorate of this vegetable research, BHU was not a partner, so it remained. But many of the private sector, they collaborated with us for screening their material, their parental lines for this thing. So in this way, we helped them to identify some of the lines which were because we were having the all this methodology standardized. So those who were wanting their line to be evaluated, we evaluated on the this payment basis.
Happy to hear that, because that is APP that is missing sometimes from the ICR system.
Yes,
But I am glad BHU is doing that, because we know for sure in tomato, and also in, as you showed in mustard also, that there are hybrids that have, see, in tomato, unless there's TLCV resistance, even if it has high heat tolerance, you cannot grow them.
You cannot do
because of the virus.
Yes,
right?
yes, very true.
So
Very
you
true.
need to combine it with the other seeds. I think the question I have is, what you talked about the fact that unpredictable environment is now becoming more common than, it's more often that we suddenly get a rain, suddenly gets a bigger temperature, and suddenly there is water stress release because we can irrigate. So there is system like plants need to have resilience against different conditions in the same time. So how do we select for that resilience?
Sir, there we have to create we have this for, I would say for mustard, we have developed the system like rain out shelter. We are screening 4 conditions here. Not only for this rain-fed condition, not only for high temperature, we are also screening material for submergence condition.
Okay.
Because of erratic rain at
Yes,
the time of
exactly.
sowing, germination is we have seen our material this year was washed off because of this torrential rain in October. So we are screening the material for different conditions. 4 conditions we have for abiotic stress and 2 major diseases like your white rust under late sown condition and alternaria blight and one for this downy mildew. So simultaneously, if we are making the selection of a panel, we can come out with some of the genotypes which and we came up with 2
Genotype.
drought as well as high temperature stress. Under a UPCAR project, we come up, we identified around 5 to 7 genotypes which were having, they have been released for yield trait attribute. They have not been screened for these particular trait. And now UPCAR is saying that you register these, re-register these lines since they are only for the yield attribute. I'll say that an RCHB 101, your RH 402, 406, these are the lines which are having both the stress tolerance, high temperature and the drought stress. And they need to be either re-registered or this thing so that the farmers can be benefited. They are having the stacked 2 stress tolerance. And also I have found that if you are working for the heat stress, it is also having confounding effect with the water stress also. And it is better to create a condition, one rate out shelter, one your high temperature late sown condition, early sown condition, and submerged condition.
So simultaneous screening is important.
Simultaneous breeding is required.
Because that is not done.
And over 4 years with the Rapeseed-Mustard, we have come up with these facilities.
Okay. Thank you. I think that's what I think future way forward is, that you have to identify those genotypes. They are there, but unless we screen, we won't know. And we may have to create those artificial screening conditions where the same set is tested against those 4 or 5 different environmental stresses, right?
Yes, sir. Yes, sir. Yes.
So that's a line of work where emphasis needs to be put, and hopefully there will be funding by the appropriate agencies. That is, there are some other hands have been raised. Anybody wants to ask a question? There was a who is that?
Vikrant Patil is there.
Vikrant Patil, please ask your question.
Yes, sir. Sir, first of all, congratulations, sir. Very nice, informative presentation you given for all of us. So thank you. Sir, my question is, are you started any projects on GPS or GWS for the mustard?
Sir, we have started this thing. We did the phenotyping. Now GPS, this is again with the UPCR project. We have collaborated with the Mahesh Rao in IARI in an RCPB, who is going to follow the GPS those genotype which I have identified in the field and this controlled condition, they are going with the GBS at IARI, and they will be giving us the data. So it is a preliminary stage, so that I would like to update you.
Okay, sir. Okay. Thank you, sir. And sir, other than heat tolerance, are you covered any other traits, sir?
Yeah, this year a novel trait we are identifying with the submergence or water logging, you should say, because we are having a big area over here. Those are the canal irrigated, and the water remains even in the Kharif season. If you sow something, so the crop will be delayed. So the farmers are going for the direct seeding and to capitalize the residual moisture in the paddy field. And there we require submergence tolerance in the early stage. So those canal irrigated areas are our target area. And we have come up, we found that most of the genotype, they are killed, but some of the genotypes, they remain in the field, and they do give a good germination percentage as compared to the sensitive one. And later on, so we have given 2 stages of submergence, one at the sowing stage and other at before flowering at the vegetative stage for not less than 7 days to 10, 15, 11 days. 11 days for germination, and 6 to 7 days at the before flowering stage, vegetative stage. So we found that the response is different at both the condition, and we found that there are certain traits which I will I'm in publication mode. So after this publication, I will tell you how to go with the submergence tolerance also. And we have gone with the rain-fed condition, rain-fed area, this Pada and all, this area is there, which is under rain-fed condition. So we are also breeding for rain-fed condition. And
Thank you.
the next zone is our primary objective.
Thank you so much.
Thank you. And there is one Madhu Banot, you want to ask, or shall I read your question? Madhu Banot, are you there? There was a question: to what extent are double haploid techniques used in accelerating heat-tolerant line development in rapeseed-mustard? And which breeding strategies have proven most effective for pyramiding heat tolerance with other agronomic traits? Big question.
Yes. Double haploid is one technology which is very good for speed breeding.
Yes.
You can diplotize in a single generation, and you can have a greater viability, just like F2, and you can make the selection over there. And the thing is that the double haploid in mustard, only Delhi University Akshay Pradhan group is working on that. And they have come up with a good this thing, and they have mapped QTL also on the basis of double haploid in Jungshia. And it is a good approach, but since the technical feasibility is not there with all the institutions, so that limits us. But we do go for this off-season in Wellington as well as in Himachal Pradesh. We are taking, our students are going over there, and they are coming up. They are taking 2 generations in 1 year, and they are advancing the generation.
Thank you. I would like to add to that in mustards, not only mustard and canola, besides that, cauliflower, cabbage. Those have been the first crops where we are commercially using double haploid.
Yes.
On a big scale. It has been one of the easier crops to do double haploid.
Yes.
Now, of course, hot peppers is the other crop where double haploids are used.
Yeah.
And I think pyramiding, of course, pyramiding will depend on the hard work done by the breeders by putting it
Yes.
all together and then selecting. I think there is a compliment by Abhinav Geware saying that his MSc thesis is on screening of Indian mustard for thermotolerance and seedling and terminal stages. This session has significantly enhanced his understanding, and so it was very for that I will put.
Well, I'm grateful.
I am happy to say that for that you deserve a cheers. So thank you very much. I think anybody else quickly.
Yeah, Madhu Banot is asking something.
Yeah, same. That was a question from Madhu Banoj. How effective are CRISPR-Cas9 and other gene editing tools in improving heat stress?
Yes,
Okay.
for glucosinolate tolerance, we are Naveen Bist has to be given big clap that he has come up with the gene editing in mustard for the glucosinolate in the seed only remaining part because of the transporters. They have come up with a gene edited Indian mustard. Yes, we can follow this technology with this, but before that, we have to identify there are several QTLs. We have to know your transcriptomics. You have to
Yes,
know
exactly.
the way, what are the pathways which are involved and which need to be blocked. So that, because you cannot make edit all of the QTLs, all of the loci, you need a single candidate pathway, single candidate transcriptome, which need to be blocked. So a vast study still is to be done on this area.
Yeah, I think I would like to add that is the reason why the two rice gene edited varieties have been. They found the knocking out cytokinase.
Yeah, very good.
Oxidates. Because of that, there are multiple things happening. Once you knock it out, it becomes drought tolerant. It has had salt tolerance. So it is these kinds of things have been reported, but now they are specifically able to knock out those, like, because when you knock out, these are something like master gene. When you knock that out, other traits start showing up. So I think it's a technique that will gain importance in the next 10 years, much more, because
and we are again hopeful that we are going to get for abiotic stresses also. They are gene drought responsive, this abscisic acid responsive gene, RAB gene, DRAB gene, which can be knocked out for, say, drought tolerance or like that. So same way the heat can also be taken care of.
Once they identify the
key pathway, yes.
Yeah, then they can do it. I think that is where it will lead to. Next 10 years are going to be moving towards gene editing. I'm absolutely sure. There is any significant genes identified for sclerotinia resistance?
Sclerotinia tolerance and alternaria. These are two alternaria that is it is not having any resistant source in cultivated background till date. Same way, we are not having the resistance for sclerotinia. Both are traits for which we are not having resistance. Hisar, they have come up with that HAU has come up with one line H228, which is sclerotinia tolerance. They claim that it is sclerotinia tolerance, but it is not so. But we have got some this national panel that is by Punjab Agriculture University. Those are few of the genotypes are showing not complete resistance, but moderately resistant, they are showing, and those we are using in transfer of the sclerotinia tolerance in our material. And one EC line, exotic collection line, is there in, I'm not having the exact number right now. I will tell you, that is giving a good level of sclerotinia tolerance. So we can identify the genetics, and one of the student is working on that genetics, and better to go with the GBS and this technique and transcriptomics to find out the resistance pathway.
Thank
So
you.
we are on this. Our students are working on this.
And thank you. I think we have Dr. Mohan, you wanted to say something?
Yes, I would like to congratulate Kartikeya for
Sir.
not only exhaustive, but extremely exhaustive, super exhaustive
Thank you, sir.
presentation. And the reason is that you have combined two crops,
Sir.
tomato and mustard.
But
So it's-
similarities of expression are there. So it's
Yeah,
good.
that's what I'm saying. So the carry home message was actually a bit confusing for the people who are not dealing in those crops.
Sir.
Another
I think-
observation, so far just now Tikoo mentioned about the GD rice varieties.
Mhm.
Can we expect some surprise from a university like BHU in the near future?
Sir, our team is working, sir. Hopefully, they will go in wheat. They are working. Our
Yes.
Dr. VK Mishra group is there. They are associated with the CMIT and all. So they are one of the student is there. He's working on editing with some of the resistance to some of the spotch plot and all. So hopefully they will definitely come. But with my work, still I have to look into.
It will take some time.
Yeah.
So we have
Okay,
Tahinyat
look forward.
Shana also raised hand. Tahinyat, are you there?
Sir, pardon, sir, I couldn't.
Just tell me, there is one Tahinyat Shana, which I see has raised hand.
Okay.
I don't see, is it a lady or is it a male? I do not, because I can't see the photo. I can't see the video.
Because there are a few questions which are
Okay,
coming from the
okay.
YouTube.
Okay, there is one question here. We have 5 more minutes, so I request everybody to be there. Which hybrid tomato can tolerate more heat? So you have shown Sadabahar into Florida.
Yeah,
717.
Fla 7171 and Pusa Sadabahar has given good this thing, and this Pusa Sadabahar and one more.
And also
Azad
your
Afri or something,
cherry
they are
into-
of a good fruit size.
Cherry into Superbug. I think the message there is that you need to have both parents heat tolerant. You will
Yeah.
get better
Yeah.
F1 heat tolerance.
Very true, very
And
true.
he says, which GM line has better heat tolerance? I don't think there is any GM work going on.
Any
We will
GM
not talk-
for this thing.
We will not talk about GM.
Yes, sir.
Gene editing
Very
पहले होना है,
dangerous to talk.
Yeah. फिर GM की बात
Dangerous to
करेंगे.
talk about GM.
Is there any change in the phenotype of heat tolerance intolerance in tomato plant? Any
Sir, Sir, there is as such, I cannot say there is any change. What I find only, see, relatively under normal condition and heat stress condition, there is a fall in the fruit size. There is a fall in coloration, a patchy coloration appears. There is a fall in the number of fruit set. So these are the variations you will invariably observe in
susceptible ones?
two conditions.
Yeah.
But as such, the heat tolerant hybrids or the variety, they will be having better seed fruit set, better size in comparison to the susceptible.
I can add to that my few suggestions. Generally, if it is heat tolerant, the leaf roll will be less, and the plant green will be a little more stay green than
Yeah, it is there. It is in tomato we observe. We
in
observe this.
tomato.
Leaf
That heat
rolling.
susceptible one, almost even the leaves don't develop very well. It's
Yeah.
kind of becomes very straggly. Whereas the good one will have, and especially if it has TLCV tolerance, leaf roll is almost like very, very minimal. There is
Very-
still some, but it is minimal. Okay, I think that. Anybody else? Shall I conclude? Give me the permission.
Yes, sir. It's 2 minutes remaining, so please.
Thank you. So I think, first of all, I thank you again. And despite my friend Dr. Mohan's observation, I would say you covered both the topics within the time limit very critically. And I think many of the things are common across. And I think my takeaway from today's talk is this. And I think with that, I would like to summarize. It is clear now that we have to breed for resilience, not just drought or not just heat or not just submergence or heavy rains. So, and also blight tolerance, foliage tolerance. So we have to combine a lot of stress tolerances together, and then you may not find all these traits together in one or two genotypes, right? So you may have some where seed germination is good. You may have some where early vigor is better. You may have some genes where fruit set is better. Pollen germination, all the viability things you said. So there are a lot of complex traits and they may be distributed in genotypes. The key now lies in us finding those from all our germplasm collections, whether within the cultivated species or the crop wild relatives. Once you have identified those in different traits, we'll have to find the either transcriptomes that are responsible for them or the sequences that are giving you trait-linked traits. If those are found, then we'll have to find a way of pyramiding them into genotypes. And then if you are breeding hybrids, you will have to divide what to do in female and what to do in male. So a complex thing, but I think this beginning has happened, and I'm sure in the next 10 years, we'll see a lot of resilience breeding, not just drought or heat or anything, because plants will start mimicking what we have as human bodies. We go to 40 degrees Celsius, we are surviving. The same person goes to Ladakh and -12, -17, we survive. So we will have to find that resilience within the QTL system that is available there. Thank you again, Dr. Srivastava. Very, very nice talk. I think a lot of us have learned how to deal with this. You can all express your feelings with your emojis that are there in front of you. And thank you very much. Till we meet again. Aparna, any closing?
Yeah, well, thank you very much, sir. And as Tikoo sir mentioned, that you are one of the very exclusive speaker who finished the whole presentation exactly on time, and we have plenty of space for the discussion. And it is really engaging. I see a lot. Last question is coming from Nigeria, but I will pass you that question so then you can give the answer later.
I will answer.
And that's the purpose of this webinar, and we're really able to make this purpose. So thank you very much for this informative presentation, and we are looking forward to give all these questions to you, which we have received from different platforms. So I'm sure you will revert back. And thank you all the participants for joining the session.
One
Yes,
minute. I want to reply Caleb Onat, you can, I think they are from Nigeria.
Yeah.
Please understand today the best solution for tuta absoluta is IPM. Please go for the biological control. It has worked very well. Since 2014, I have had no problem of tuta because I plant every 15 IPM those traps within an acre at that rate. Go to a biocontrol company that is doing that. It will help you that. Breeding will be a long term. It's too complicated. There is Solanum chene. What is that? Chinensis. I think that is LA711. resistant to almost like 7, 8 different insects, you can try that. But that's a long shot. Will take you. Thank you very much again, everybody.
Yeah,
And any questions, please, Aparna, you can send it to Dr. Kartikeya.
definitely, definitely. Thank you,
Goodbye.
everybody.
Till we all meet again. Bye bye.
Thank you, sir. Thank you.
Good day to everybody.
Thank you.
Well,
Thank you, sir. Thank you.
Thank you.
Thank you very much, sir.